Okay, I think it's good time to get started. So, hello everyone. I'm Sai Lakshmi Subramanian, Program Manager at Seven Bridges. On behalf of the Cancer Genomics Cloud team, it's my pleasure to welcome all of you to this Proteomics virtual workshop. So, the Cancer Genomics Cloud, CGC, powered by Seven Bridges, is one of the three systems funded by the National Cancer Institute, NCI, and we are very honored to be part of the NCI's Cancer Research Data Commons infrastructure, which is CRDC. CGC provides a secure and scalable computational cloud-based platform that facilitates analysis of massive public data sets like the TCGA, the Clinical Proteomic Tumor Analysis Consortium, or CPTAC data sets, and several others. So today we are here to learn about how to use the features of the CGC to perform proteomics analysis using data from the Proteomics Data Commons, or the PDC. I'm very happy to introduce the presenters of today's workshop, Mr. Dave Robertson and Dr. Felipe de Vega Leprovo. Mr. Robertson worked for 10 plus years at the NCI's Intramural Cancer Genomics Research Lab as a wet lab assay developer and as a bioinformatician. He's currently in the role of community engagement specialist at Seven Bridges, where he leads trainings, develops tutorials, and engages research groups in cloud platforms such as the CGC. Um, Dave has run several workshops and hands-on demos for users of our public programs platforms, including CGC, the NHLBI Biodata Catalyst, and Kids First Quad. I'm also delighted to introduce Dr. Felipe de Vega Leprovo. Dr. Leprovo has a dual degree in biology and systems analysis. He holds a master's degree in cell and molecular biology and a doctorate in bioinformatics, both acquired at Fiocruz in Brazil. Felipe currently works as a research faculty at the University of Michigan Medical School. He's a member of Professor Alexei Neshvitsky's Proteome Bioinformatics group, where he develops new analysis methods and software for proteomics. So before we start, I would like to offer my heartfelt thanks to the NCI, our program officers, Dr. Erika Kim, and also members of the PDC and CPTAC team for providing guidance. A special mention to Dr. Sudha Venkatachari, Rajesh Tanguru, and Paul Rudnick for providing guidance to organize this workshop, and most importantly, all of you attendees for supporting this today. Special thanks to Dr. Dan Ventry from Seven Bridges for coordinating the announcements and other logistics of this workshop. So without much further ado, over to you, Dave and Felipe.
Thank you so much, Sai, for the wonderful introduction. So welcome to Unraveling the Mysteries of the Human Proteome, from tools to workflows and analysis. My name is Dave Robertson, and let's go ahead and get started with an objective. So the objective for today is going to learn about the capabilities of the Cancer Genomics Cloud powered by Seven Bridges while analyzing the proteome of clear cell renal cell carcinoma samples. And we're going to focus on the integration, the tight integration with PDC. Please stop us and participate, ask questions throughout. We have 2 hours. So let me talk about the agenda, but there will be a break after the slides for 5 minutes. And then we could also build in another 5-minute break in the second.
Dave, yep. Just wanted to announce that we are recording. I forgot to make the formal announcement.
Yes, we are recording, and we can distribute that later for you to see. So if you fall behind, you can always watch the recording again. Great. So for the agenda, we always like to start off with a scientific question to frame the workshop. So we'll give some background on proteomics and ccRCC. Then we'll do some slides that talk about specific Cancer Genomics Cloud platform features. Towards the end, we want to have an announcement about some multi-omics webinars that will be coming up this month, actually, and also really emphasize how to get support after the workshop. So we're all about engagement with the users, and there's a tremendous amount of ways that you can talk to Felipe or myself, as well as our support team at Seven Bridges. We'll have a 5-minute break right after the slides. This will actually be a good time if you have not made a a CGC account. They're free to make, and you can easily make one with just your email address. So that'll be a good time to, do it on the break if you need to make one. But I think plenty of people already have accounts. And then most of the time it will be spent in a live demonstration. It's hands-on from everyone that wants to participate. But the way we've set it up is I don't want you to feel pressure that you have to follow along at every single step. You can reach out to us later today, tomorrow, or whenever you have time to complete the project. So let's go ahead and get started. So I mentioned some acknowledgments. We are really grateful for everyone on this slide, the Global 7 Bridges team, you guys, the CGC users, and all of the collaborators, the Novichky Lab at the University of Michigan, the PDC team, as well as the other CRs. So there's a tremendous amount of collaboration in this ecosystem. Okay, so let's get going. We always like to frame stuff in a scientific question. So today's question is, how do I analyze the spectra data of the proteome in clear cell renal cell carcinoma cases? Now, I'm not a proteomics person. I worked in a genomics lab for 10 years, but proteomics is new before this project is new to me. And I think we have two groups of people on the call. So we have proteomics experts, and then we have maybe Cancer Genomics Cloud experts, and maybe a mixture of both. So everyone can participate in answering the scientific question. So as you know, kidney cancer is among the 10 most common cancers in both men and women. So what we're going to do is we'll demonstrate going to the Proteomics Data Commons and finding the CPTAC data. It's multiplexed, and it allows for differential quantitation across multiple tumor and normal samples. We're doing the first stages of the characterization, and we'll get more into that later. But frame everything, we're focusing on kidney cancer and how do we ingest data from the Proteomics Data Commons and start analyzing it in the CGC. Okay, so that's a big picture. So this is the typical user flow. It starts with logging on to the Cancer Genomics Cloud and creating a project. I've already made a project that we can all use. Each member that I had their username, I went ahead and cloned them a copy of the public project for this webinar. During the break, let me know if you don't see that project or if you're a new user, and I'll make you one. But the flow is you make a project, you select your data by multiple different ways, and we're going to focus on one way today, which is ingesting data from the PDC. Then you select your tools from the public apps gallery. And finally, you run your analysis, and you can do interactive analysis in RStudio or Jupyter Lab, or you can use cloud scale batch jobs, which we call tasks, which we'll talk about later. So let's refine this. Today's analysis flow is all attendees that want to participate, log in and review your workshop project. And I will demonstrate how to make a project because it's a very important concept, but yours is already made for today. Then the workshop project's already been populated with the PDC data, but we will demonstrate how to ingest that data if you want to repeat this project or if you want to try a different disease case. That'll be very useful. So we'll demonstrate that later on. The tools, most of the tools have already been added to your workshop project, but they are in the public apps gallery. So in one of the steps, you will get one of the public apps and use it. And finally, we'll run some tasks. We'll do an interactive analysis in RStudio, and we'll also run some batch tasks. So let's talk about projects. I just want to briefly talk about these different categories and touch on some things. So projects are the main organizational unit within the Cancer Genomics Cloud, and just think of them as containers. They're containers for files. They're containers for apps, which are also known as CWL tools. More on that later. And when you run a CWL tool, that's called a task. So a task is either a running analysis or one that's already completed, and you'll see examples of that. So here's actually a screenshot of the webinar project, which I cloned one for everyone. We'll review this later, but on the left side, it has a description of what we're doing, and you'll see project membership. So I'll be a member of your project and help you during the workshop, and also later on, you can message me through the project actually. And then you'll see some introductory analyses, and then we're going to add some more analysis on top of that. Okay, so everything starts with the project creation, and then here's all the contents. So a project has 5 main sections. The dashboard is what you see here, which has a description as the members of your project. But then the next tab over is your files. So the files are populated with PDC raw files. Felipe will talk more about this soon. It also has a copy of these slides. So everyone always asks us for the slides. It's already there for you. And then it actually has 4 workflows that we're going to use today in the apps section. So you can go from files, you click on the apps, you'll see the different CWL tools to run. And then it actually has some pre-run examples in the tasks. We're going to add some additional analysis on top of that to increase the overall experimental size and number of channels we're analyzing. And finally, way over on the right side, if you can see my mouse, is the interactive analysis. So this means that we have RStudio, Jupyter Lab, and you have those 2 options to interactively analyze or make visualizations. So let's dive in a little bit deeper. So let's talk about data. So there's a lot of diverse data types that can be co-analyzed in the CRDCs. And this is the slide we always present here. There's many different data silos. So we have proteomic data, genomic data, integrated canine data, and imaging. So the whole concept of this ecosystem is that you'll be able to log into one of the CR platforms and link the data without actually moving it. So build a cohort, link the data, and then analyze it. And there's contributors and consumers. We like to serve biomedical researchers, tool developers like Felipe and myself. It's good for tool developers because essentially everyone on the workshop is going to be testing this tool today at scale, as well as data scientists. And there's lots of ways to interact here. So the key integration we're focusing on and why everyone is interested in this is the integration with the Proteomics Data Commons. Actually, Sai, do you want to mention anything about the Proteomics Data Commons here? I want to make sure we do it justice.
Absolutely. So Proteomic Data Commons is one of the nodes within the CRDC, which aims to democratize access to all proteomic data sets, or anything cancer related to anything cancer related. And you will be able to go to the website, PDC website, and there's a data portal. So if you are generating proteomic data sets, you can submit your data, and all the data is processed in a harmonized way, and it's available for further analysis in cloud resource platforms like the Cancer Genomics Cloud. So you can access the data within the user interface or also using their APIs. And as you can see in this bottom figure, there are over 62 studies, over 24 terabytes worth of data in the PDC. And I want to highlight that the CGC integration with the Proteomic Data Commons, it has enabled tool developers, like what Dave pointed out earlier, and researchers to take the advantage, full advantage of the cloud resource, accessibility to scalable cloud resources. And what we have found with this integration is that proteomic tools can run very quickly and efficiently in the cloud, so which lowers the barriers for usage. You don't have to rely on your local HPCs, but you can run everything on the cloud in the context of your own data sets or publicly available data sets.
Thank you so much, Sai. Okay, so we're going to demonstrate this flow during the hands-on part, but this is a slide which indicates how the integration was facilitated here. So the idea is you go to the Proteomics Data Commons first, you build your cohort. So we'll do that with CCRCC data. That part's just a demonstration. And then you download a manifest of the files you want to import. You go into your project and you import, you go to import a manifest file, you upload that file through the web browser. So you can see some of the different options here on the right side. And then the import is very fast. So essentially it's linking the file objects from the PDC bucket to your cloud bucket. And there's symbolic links there. So there's no really heavy duty moving of data. So that's one of the benefits of using the cloud resources. Okay, some of you I noticed are TCGA researchers as well. So we have a great way to query the TCGA data, which is the Genomics Data Commons, on the Cancer Genomics Cloud using the data browser. So here's, and this one looks a little different. It's a visual query. So you can start with all approximately 11,000 plus cases and filter, through the kidney cancer investigation. And then you can find samples, in this case, primary tumor, which matches the solid tissue normal, and then different types of data types. So the theme is different multi-omics data types. So here we can find gene expression, quantification, and methylation beta value. So these are actually, I just know that these are open. So you don't actually need, in many cases, you don't actually need controlled TCGA access to access some of these files. But if you do, you can go back to, I think, the higher levels or the previous levels where you can get, the more controlled forms of the data. Okay, so we've talked about projects, data. So now let's talk about tools. And this is where Felipe is going to help me out a big time because he's the primary developer in the tool that we're featuring today, which is called the Frag Proteomics Pipeline. Okay, so I'm going to pass it over to Felipe for the next couple slides.
Hi, thanks, David. Thanks, Sai, for the introduction. So in the next slides, I'm going to introduce a little bit about the pipeline, the Proteomics pipeline that our group has been working in the past years. And what I'm going to show is actually one of the main tools and central tools for the proteomics data analysis that was featured in a few of the publications from the CPTech, the third edition of the CPTech. More specifically, what we're going to show today is how to execute the pipeline in a way that you can actually reproduce the analysis that was done with the kidney data, the CCRCC data that was published a few months ago. You can go to the next slide, please. So I noticed that some of you are actually have never used a proteomics pipeline before. So I added this slide to go briefly over how the data analysis actually works. So in a very similar way to the genomic analysis, the proteomic analysis happens in steps. And in each step, we process the data in a certain way that it allows us to filter and quantify the peptides. So, so basically what the pipeline is doing is we start with the database search, more specifically using a tool called MS Fragger. So MS Fragger is a ultra-fast database search engine that we published a few years ago. It actually allows you to, it's actually so fast that it allows you to do what we call open searches, which is the ability to do a database search without restricting for delta masses. So the database search is the first step, which allows you to match the spectra from mass spectrometers to a protein database. The matched peptides are then validated. That step is called the peptide validation. We have a tool called Peptide Profit that was inherited by the TPP set of tools that we incorporated into our pipeline. All validated pipelines are then used for the protein inference. For the protein inference, we also use another TPP tool that we incorporated called Protein Profit, which gives us a set of proteins built based on all peptides with high quality. And then we apply the usually false discovery rate to the data set to allow only a percentage of high quality peptides, ions, and proteins to be considered. And after that, we use different strategies to quantify the data. So what we're going to show is a workflow that incorporates the label-free quantification, which is that consists of using the spectra intensities from the spectra that the instruments acquired, and we are also using the intensities for the TMT tags, which is basically a barcode that we incorporate into the samples. So basically, that's a basic workflow that most pipelines follow. It's pretty standard, and we basically have a tool or a set of tools that can easily give people access to these all these options in a very easy way. So next, please. So those are the steps. This is just to show which tools are involved into the analysis. So here we're basically showing 3 of the Frag pipe tools. Frag pipe is actually a larger set of tools that allows you to do all kinds of analysis, DDA, DIA, team stuff analysis. But here we are basically showing how you can use MS Fragger for the database search, Philosopher that orchestrate and organize all the other tools, and also allows you to do a very stringent FDR filtering and the quantification. And then we finish with the TMT integrator, that is a tool that does a statistical summarization and reporting at different levels, like a gene level and a protein level. So all of these tools are published. They are either published or available in our GitHub page. Anyone can use. They are free. We don't charge for people to use them. FragPipe is MS Fragger is also free for academic usages. And I encourage you to visit our GitHub account. There's a link over there on the corner. And check what we have there. We have a very responsive team. So if you have questions, if you want to ask for some kind of a feature or anything like that, you can visit our GitHub account and reach us and get in touch. And there's also another link on the other corner that will take you to our to a discussion board that we open on our GitHub. So if you have any questions during this workshop, you can go there right now and just post your questions, and me or one of my colleagues will make sure that we have you covered.
Great, thank you so much, Felipe. Oh, can you talk about this one as well? Elise handles.
So this is one of the interesting things about what you're going to show. So for the CCRCC paper, more specifically, the kidney cohort is actually made of 25 different experiments. Each experiment consists of about 10 patients or samples, or in this case, about 10 different tags. So we have approximately 500 samples that we need to search, validate, quantify, and filter. So that's a pretty extensive. That's a pretty considerable amount of data. For the paper, for the project, we use a local server that it was provided to us by the University of Michigan. That's a blade server with 56 Xeon cores, pretty high efficient device, about 500 gigs of RAM. And for the analysis, it took us about 27 hours and 16 gigs of RAM. That's actually already something pretty impressive, because most of the pipelines or the other tools that we are available right now, they cannot reach that results in that amount of time. And the analysis that David and I are going to show you is actually it's using the same data, but it's actually much faster. So the way how the CGC workflows they operate, and the way and all the infrastructure behind them are extremely tuned, and they were designed in a way that they can actually provide an infrastructure that is extremely efficient for this kind of analysis, and it can give you results in much faster than what a high computing device can. So you want to talk about the public apps?
Yeah, absolutely. I muted myself by accident. Okay, so yeah, thank you for that background, Felipe. So the tools we're going to use, you can find today on the public apps gallery. So as soon as you log in, you can click on the, we'll show you how to do this. You can click on the public apps gallery and search by category. So in this case, I'm searching by actually two things. There's more than just Frag pipe for proteomics, but if you click on the proteomics category and then the Frag pipe toolkit, you'll see these 4 workflows, and we'll talk in detail about why there's 4 of them. Thanks. So essentially, you can think of it as one large pipe, but we broke it up into 4 sections to actually increase the speed of it, because there's certain times where things are very parallel, and then there's certain times where all the data comes together and is merged. But there's also over 500, I think it's 574 bioinformatics tools and workflows that are optimized for the cloud. So if you're doing multi-omics and other things, check out what's there. I think now we're going to break it down. Yes. So inside the project, there's the 4 workflows. And actually, I left the 4th one out because I want everyone to get practice cloning a workflow, the last step, the TMT integrator in QC that Felipe was talking about. But you have these 3. So the first step is to do MS Convert, MS Fragger, all the way through Peptide Prophet. And then all the data comes together for Protein Prophet, and Felipe can feel free to jump in whenever. We'll probably get more into the details later, but everything comes in together during Protein Prophet. And then we split out the workspaces again on the 3rd step to do filtering, quantification, and reporting on the protein abundances.
Yeah, that's right.
And then the final step, everything comes together again to do TMT integrator and a QC. Now, quality control is a notebook, an R notebook that Felipe made. So we're going to run the R notebook as a tool in this workflow, but also interactively in our studio, so you can customize it. And we'll show you how to do that. So I always like to look at these. So these are directed graphs that represent the analysis flow. So there's 4 of them. And then the first one is Frag pipe that takes in the raw files directly from the PDC. So what happens is just linked to your project. It's when you hit run, it actually goes to the PDC bucket and then starts to ingest it for real. He mentioned that there's an end user level agreement. So if you're an academic or using it for educational purposes, non-commercial, you have to accept this. And we have the whole user agreement in the description, but that's just because of the MS Fragger license. But once you accept that and you include the raw files and the reference database, you can go ahead and run. So there's the default parameters have been filled out by Felipe, but if you want to. Honestly, there's a tremendous number of parameters. I think more than any other workflow I've seen. And Felipe added hundreds of parameters that you can that have been documented. You can change. So that's the first step. The second step is more simplified. It takes every workspace and at the top level does protein profit. And you get one file, which is a protein.xml file. And then you take that output, and then again, we're going to use the cloud multiple parallelization to add in annotations, to do philosopher filter, label quantification, free quant, and then finally report. And then finally, everything comes back together in TMT integrator. Okay, so this is all made possible. All the development was done on the platform and completed very recently, and now we're sharing it with you. So you have Web Composer in each of your projects, which gives you the ability to tailor these pipelines. So that's what we use to build and test it. It's intuitive, flexible, easy to use. So even if you're a coder, you can still use this. And I think it's really, that easy to use. It's built on two things. Now, we asked you if you're familiar with Docker. About half of you said yes, and the other half said no. You don't have to, we're not going to cover making Docker containers today, but I just want to briefly mention what they are. They're portable. They are portable compute environments. I was about to say Linux environments, but we actually, I guess you can have a Windows Docker, I suppose, but today we're going to be using all Linux, portable Linux containers. And what they do is they allow you to encapsulate all your software dependencies, including certain versions of different tools. And the common workflow language allows you to run them at scale in the cloud, and also to use programs like Web Composer to tailor them. So, example of tailoring may be something simple that you might want to do. So TMT integrator actually creates a list of files as the output. You may want to actually compress those into a zip file to download or send to somebody. So you may, you may create an archive. So there's a public app to create the zip file. So that would be a simple example of tailoring a workflow. So we're just about over with the slides, and then we're going to do a break. But the final step, after you've selected your tools, is to run your analysis. So I want us to run 4 cloud tasks using the 2 additional plexes. So Felipe briefly mentioned that there's 23 plexes or experiment names, which contain samples inside of those. So this is a great way to batch things. So I've already ran the first 21. So we're going to add an additional 2, because I want to get you guys to have a feel of how to run multiple experiments in parallel. And then we're going to use RStudio to interactively quality control and cluster the results. Now, every step, the results are already in your project. So, that's how we were able to compress this. So it won't quite fit. The entire analysis won't quite fit. It's about 4 hours to go from start to finish. We only have 2. So that's why you can get the first part started, and then the resulted files are already in your project. So you can go ahead and start the next one. So a great feature about the analysis, if you decide to use the common workflow language tools, is you can monitor the analysis at a macro or micro level. So a macro level may be what's in your project. You click on the tasks tab, and you can see what's running, what step it's on, what were the inputs, what were the app settings. So there's a tremendous number of default app settings that Felipe has coded, so you can expand these and monitor those. You get to see the duration, and also how much of the cloud credits were used. By the way, we're all going to be using a training cloud for today. We're all going to be using a training billing group that Sai set up for us. But if you want to drill down and look at the micro level, you click on a button on the task, and it shows you a timeline of all the events taking place. So you'll see, can I zoom in? Oh, I can't zoom in there. you'll see multiple, so each line represents a job. So there are certain times that it can run jobs in parallel, and certain jobs like MS Fragger take a medium amount of time. And then you can see that this step peptide profit took the majority of this particular workflow. So that gives you an idea of how long each step took, which is much better than running it as one long script. So you get this overview. And you can actually drill down even further and look at the individual log files of what's happening in real time if you want to. So this is the key point I want to tell you guys about. You can scale up by batching by the Proteomics Data Commons file metadata. So when you use the import feature, not only do the file links come along, but you also get some rich metadata that you can use. And we're going to demonstrate batching by experiment. So each experiment is going to be a group of 25 items. And we're going to, well, you're going to do it with 2 of those to get an idea of how you can batch. But earlier I went ahead and did it with the remainder of 21. And then you can review you don't have to download anything. You can look at the outputs without downloading. So here's an example of an HTML report that Felipe made. You can just click on it and get a preview. Many different file types are viewable on the platform. And you can actually, invite a PI or a colleague to your project and just send them the link and say, look at this report. But if you want to customize your report, you can do that with RStudio or Jupyter Lab. And both of those are available for you inside your project. What's next? Okay, I want to briefly make an announcement that we have a Cancer Genomics Cloud monthly webinar series, which allows you to learn about a variety of different things. We just started it this year in January. So these slide decks is in your project, but if you go to cancergenomicscloud.org/webinars, you'll see the latest webinar, which happens to be, I think, really nicely related to proteomics, because they're going to talk about integrating multiple omics. So not just genomics, not just proteomics, but how do you combine them both? It's the 4th Wednesday at 2 p.m. Eastern time. If you need support, we're here for you. So there's docs at docs.cancergenomicscloud.org. You can contact support 24/7 at CGC at 7bridges.com. So what that actually gets sent to a support team, and they're basically staffed almost 24 hours a day, and they'll get you the support you need for any issue you may have with your analysis. I host, my colleague Anna and myself host office hours every week on Thursday. So if you go to this link, you can see when they are. And just stop by. You don't have to sign up, just stop by and talk about whatever, you can tell me how far you got in the workshop and what you might want to do next. Yeah, so here's an image of what the cancergenomicscloud.org looks like. There are, under resources, there's webinars and publications, releases and events. And under help, there's a quick start tutorial and a more comprehensive tutorial for how to use, controlled TCGA, for example. Okay, Felipe is going to do this slide, and then Sai has an announcement, and then we're going to take a break, everyone, for 5 minutes. But if you could just bring it home, Felipe.
Yeah, so as a final point, I mean, the message that we want to give is that the way how things are moving today is towards a multi-omics or an integration aspect. So the idea is now to face and interpret these different biological and complex conditions like cancer or other biological phenomena by integrating different omics or different technologies. And as a biologist and as a researcher, I don't want to spend time, we don't want to spend time dealing with software or different configuration steps or different workflows and libraries and tools. Every pipeline and every field has its own way of dealing with this software. So to me, something like the CGC is actually pretty impressive, because it just gives you all the tools to do that right away. You don't need to worry about buying expensive computing resources or hiring specialized people for each step or each tool. I mean, all the tools and all the infrastructure is already there. So I see that as something very impressive, because it just allows you to move very quickly through that step and get to the results.
Great, thank you so much. And the next slide is for Sai.
Yeah. So I got a request from the PDC team just to solicit some volunteers for a user group study they're doing. So there's a short survey. I can post the link there. So you can sign up to, schedule an interview. So they'll be asking you some questions about how the utility of the PDC. So just wanted to announce that on behalf of the NCI and the PDC teams.
Thank you so much. Yeah, please fill out the survey if you can. Okay, take a I've been told that there is Zoom fatigue sometimes, so let's go ahead and take a 5-minute break. So, and it'll be a true 5-minute break. So at 47 after the hour, we can come back. But if you haven't made an account, now would be a great time to do that. I think most people have, but if you want to follow along, go to cgc.sbgenomics.com and make an account, and I can make a clone of the webinar project for you.
So Dave, if people want to hang on, they can just stay, and if they have questions, they can unmute and ask or use the chat.
Yep, we're here. We're just going to pause briefly and okay, so I got some messages coming in, so let me just read them.
Yeah.
Great.
So we have a comment about the multi-omics jigsaw puzzle slide. Yeah, I like that too. It's from the CC paper, the proteogenomic analysis paper. So it's an interesting way to look at how omics is a puzzle and how everything needs to fit together to answer complex questions.
Hi, this is Kritika from Georgetown. Can you all hear me?
Yeah, Kritika, go ahead.
Can you hear me? This is Kritika from Georgetown.
Yes, Kritika, I can hear you.
Yeah. I had a question for David. So the project that he shared with us had you mentioned the task for identification of the peptides was not a batch job, right? Would you also be able to share a task that you showed where you set it up as a batch job? I think having that task shared also will be helpful to understand how to run a batch job for the first step.
Absolutely, and we're going to get into that in just a moment.
Great, thank you.
Great question, Kritika.
Thanks.
And I think we have one more user's question, which Kritika had answered. Question was from Steve. Can you convert a workflow on the CGC to CWL, JSON, or YAML files?
Absolutely. Hi, Steve. Nice to hear from you. I'm glad you could join. Yes, so you could, what was your question about? So there's two versions of CWL, of course, and there's YAML and JSON, and we can talk about both in a moment. We have actually can you convert a workflow? Yes, so you can export. And we actually, there's a more advanced way to do it now. So our colleague Kaushik went ahead and created a utility called SB Packer, which allows you to pull down and version control your your code as YAML, really nicely formatted YAML files.
Thanks, Robert. Great to hear that.
Okay. The person who asked me to clone a project for them, I just did it. It just took me a second. All right, so it's been 5 minutes. Where is everybody? I lost my screen. There you go. All right, let's get started, everybody. So, okay. So I'm already logged in, but if you're not logged in, go ahead and click on cgc.sbgenomics.com, which will take you to the login screen. And there's two ways to log in, and you'll know which one to use. There's you can link your ERA Commons ID, or you could create a combination password and email address. I am not logged. Wait, am I logged in? Let me hit refresh. Okay, so here is so if you're brand new, I just want to give you a really brief orientation. On the left side, so this is what the platform dashboard looks like. So we're not in a project now, we're at the top platform level. And on the left side, you see the most recent projects I've been working on. So I cloned a bunch of projects. That's why you see they're all kind of the same thing. I cloned them, and I usually put your username ahead of that, so you can tell which ones, I can tell which ones belonging to yours. And on the right side, you'll see the tasks, your most recent tasks. And your analysis is broken down by tasks or data cruncher. So tasks are when you run a job. A data cruncher is when you do an interactive analysis, and we'll do an example of both today. So let's go ahead. Just doing a time check here. Let's go ahead and review our individual webinar projects. And then we'll run our first workflow. Okay, and then I think we're back on track. So your platform dashboard is going to be less cluttered than mine. So I'm in everyone's project because I want to be able to help you out later on if needed. Everyone should click on the Frag Proteomics Pipeline tutorial, and I'll go ahead and start with this one. So this one, actually, I didn't add anyone. So this will just be mine. Okay, so this is the project that we made for you. So you guys are the first to see this. We're going to make this a public template project. So along the top menu, there's different things that we'll go over, but one of them is public projects. So this is a great way to learn how to use the platform. There's different examples, and this will be one of the examples. So you guys are kind of the beta testers of this, but it will eventually be a public project, so you can start over by cloning the public project. On the left side, we added a description about what Frag Pipe is, and then the goals, and then the steps. So there's 5 steps we want to do today. Time permitting, and if you don't get through them all, it's perfectly fine. You can actually skip a step and then start the next part, or then just decide to watch and reach out to me. So let me just orientate you again to a project on the CGC and talk about how to get support. So along the top, as I mentioned, we're in the dashboard, but then there's a files tab, which has the PDC links There's the apps tab, which has 3 of the workflows in it. We're going to add another one from the public apps gallery. The tasks tab has your some example tasks that have been run, and that's where you'll see your running the jobs that you kick off. And then way over on the right side is interactive analysis. And that's a header that allows you to do different things. We're going to focus on launching our studio. In the settings, you may not see this if you're not admin in the project, but if you are owner of the project or a co-admin, you can change settings like the billing group. I want to mention that the platform makes billing really easy. So we set up a billing group just for this webinar, and everyone's a member. Each project belongs to that billing group, so we can see how much everyone used their projects. But if you're a new CGC user, you have $300 in cloud credits. If you do need to get support and as soon as possible, just write me a note in the project and just say, hi, Dave. And then I'll see that in my email, and I'll be able to respond in the project. Or you can also email cgc@7bridges.com. That's our support group. Or third way is on the bottom corner, there's a question mark, and you can just contact support right here. Just start typing, and they'll get back to you with whatever email address you registered on the platform. Okay, so that's enough about support. So what I want us to do is run the first workflow. So we're going to loop back later on and talk about how the data was imported from the PDC, but I want to get us started doing some analysis as soon as possible. So let's look here. Don't follow me just yet. We're going to launch an app in just a moment, but I want to show you the jobs that were already run because Kritika had a question about that.
Do you want me to talk about the question again, or
no? I'm just looking at something.
Okay.
Yeah, absolutely. Yeah, so I thought this would have been a batch task. It looks like it was just singleplex. So yeah, we're gonna duplicate that as a batch task right now. So I have 3 examples here. So the first is convert, identify, and peptide profit. So this is going to be run for each of the experiments. And why don't we go ahead and kick that off? And then the next one is running protein profit for all the plexes. And then finally, we do filter, quant, and report. So a batch task actually looks like this. The first one should have been a batch task. So this actually has you fill it out and actually has all the child tasks here, and it's all contained. Okay, so let's go ahead and launch the first job. So let's go ahead and launch it this way. So if you click on your apps tab, find the tool that's called Frag, Pipe, Convert, Identify, and Peptide Profit. And on the right side, click Run. And this will if you notice, it automatically takes you to the task tab, because what we have now is a draft task. So we're going to fill out all the options here, and then we're going to hit Run. So we're going to make sure that we turn this into a batch test, so you guys will see how that works. So if we go to, you'll see on the left side that there's 2 options for inputs. So there's the raw MZML files, and then there's a database. So the raw MZML files is all the data that we imported from the PDC. And the database file is Felipe uploaded that. It's a .fas database file. And so we're going to select both of those. Let's do the database file first, because that's easier to explain. So if you just use let me just demonstrate that again. If it was too fast, use the file picker, and it automatically knows that this should be a .fas file. There's only one in your project. So if you click on that and click Save, it'll be populated there. So now let's do the raw file. So this is the fun part. So if you click on the raw files, the file picker, it's going to list 575 raw files. So there's 23 plexes of 25 files each. And these are all in your files tab under CPTAC raw files. Okay, so this is a really important concept. You can use these options here to filter down to find the files you want to use in your analysis. So I just want us to launch two jobs as a batch task. So let's pick plex number 22 and 23. And let me see if I did that. So yeah, I did an easy way, a shortcut way to do that. I went ahead and tagged them as groups. You could also add more filters and filter by any type of metadata. So another way to do this would be to filter for the plex. But let's go ahead and click on the tags drop-down menu and click on plex 22 and 23. So I broke them into two groups, plex 1 to 21. The idea was I would run that ahead of time, and then plex 22 and 23 we would do as a group. So if you click on tags, plex 22 and 23, you should have 50 files here. And I can tell we have 50 files because on the bottom right corner, it's actually showing us 1 to 50. Okay, let's use the select all button and select all 50 files and then save the selection. Okay, but we're not done yet. We need to do actually two things. Before I forget, you have to actually go down. These have a tremendous number of, as you see, parameters here. You can minimize them by clicking it down there. In MS Fragger, there's a accept end user level agreement. So you have to go ahead and accept that. The tool will fail if you don't accept it. And by the way, don't do this, but I want to show you, if you click on any of the app name, and if you do want to read the agreement, it'll be on this screen. Once it loads, it'll show you, and this is in the public apps gallery too. It has the full end user level agreement that we have. So this is for non-commercial academic use only. So make sure you put true as accept end user level agreement. And then before we can launch this, let's make a batch task. So to do that, you turn batching on. All right, you toggle it on, and then you pick what you want to batch by. We don't want to batch by file because that would make 50 jobs. We want to group them by their experiment. To do that, we use a drop-down menu and select file metadata, which brings up all the metadata that could be populated for these files. If you go way down to the bottom, you'll see additional metadata, and one of them is plex or dataset name. If you check that and click done, it'll reformat the screen, and you'll see you have two experiments here: 22 CPTAC, CCRCC proteome with 25 items, and 20 CPTAC, etc. And they each have 25 items in them. Let me just pause there to see if everyone's following along, because this is an important thing.
This is Kritika again. I have a question for Felipe. So normally in genomics workflows, we run an algorithm that does the quantification first, and then you add the annotation. In this, even like a metabolomics, you do the quantification, and then you try to identify the metabolites. Here in this case, you're identifying it first, and then you're trying to quantify it, right? Is there a reason why you're doing it differently?
That's actually the standard workflow or standard way of processing the DDA proteomics. You know, the common shotgun data analysis. We need to filter the data first because we have to apply the false discovery rate. Otherwise, you're going to quantify peptides that might not be true positives. So that's how pretty much every pipeline operates. I mean, there are some authors that have published before different ways of analyze the data. But usually we start by identifying the peptides and then applying the filtering. And only the peptide sequences that pass the identification and the filtering receive the quantification.
All right, thank you.
Yeah.
Great. Thanks for the questions. It's really important to keep everyone engaged, I think. So that's another great question. So the idea for this first step is to batch by the plex. So we use the file metadata, and we're getting two plexes here. So we're gonna launch 2 child tasks. If you want to scale up to the full thing, you can do that later. We're all in the same billing group, but on your own billing group, feel free to scale up to the entire study if you want. It doesn't cost much money, but it does use multiple instances to get that speed. And the billing group is, each of the billing groups has a limit on the number of nodes you can use at any given time. That said, if you can actually, the system will automatically queue up the jobs. I just don't want to get any queuing during the workshop. So everything else can be default. The only required field is to accept the end level user agreement. So once you fill that out, on the right side, click run, and it'll warn you you're about to run 2 tasks. That's what we want to do. And click run. And you'll see that it's under Frag pipe convert, it's made 2 child tasks. And they're queued, and they'll automatically, we shouldn't see any queuing at this level. I like to hit refresh down here on the bottom left, and you'll see that they're already starting to run. So this is the macro level view I was talking about. If you want to zoom in, you can go ahead and click on one of these. You won't see any logs just yet, but after it starts to get running, this workflow will each take about an hour, but after a couple minutes, you'll be able to dive in and see the stats by clicking on view stats and logs. If you ever need to get support, there's another button here. You can just hit get support and start typing. Okay, so that was the first step. Now let's pivot and get RStudio up and running, just so we'll have that for later. And then we'll circle back and do the second step, which is protein profit.
Hey, Dave.
Mm-hmm.
It's Paul Rednick. I just had a quick question for
Paul.
you. Hey, you mentioned a huge number of parameters. Did you launch the task with a default set of parameters, or is there a parameter file that's linked in the background, or how are the parameters set?
Great question. So this is all built into the CWL description. So Felipe painstakingly went and filled in, typed in options for each of the parameters. And so since I'm on the screen, in any running task, you can click show all, and it'll show you the different settings which are exposed at the workflow level. So in many cases, there's even more parameters at the tool level. So we built a workflow out of individual CWL tools. So these are what Felipe exposed at the workflow level. Now, a lot of these have defaults. So even though it says no value here, that means we didn't add another value, but a lot of times you can click on the question mark to get a description, and sometimes it will show you if there was a default value. But I like to make that as clear as possible. Some of the other steps will have better options here. So a better documentation about what the default value, like for instance, like this has a default value of false. And it looks like it was filled out for a false. So yeah, so MS Fragger has a tremendous number of, I've never used MS Fragger off the platform, but it's got a lot of parameters. But yeah, so think of it as 2 different layers. So there's a tool, the parameters are built into the CWL description of the tool. And then when you add the tool to a workflow, at that point, you can say, what do I want to expose at runtime to the end user or to myself when I'm going to run this workflow? And you can change the defaults there at that level. And so we just tried to give you a feel for what some of the options are. But yeah.
Got it. So any value you see there that doesn't say no value is it's pulling it from the CWL file.
It's actually when you don't. So for instance, K-lysine here, this was typed in at the workflow level. So we typed that in to change
That's TNT.
what the default is.
Yeah.
What the default is. There could be, so we sort of if there was a parameter on the tool level, or it could probably just be, it's probably just null. You could write over top of it at the workflow level.
Hi, Paul. Some parameters, they don't have a default value. So for example, the modifications or the variable modifications or the static modifications, most of them, they don't have a default value. That's why you see a no value over there.
Okay, but how about just precursor tolerance or something very basic? Is that in there? Or?
It should be there, right? I don't know.
Precursor tolerance. Okay, so this could be, so this is version 1 of the wrapper. We can actually, I see what you're saying. Is this, I don't know if this is a required field or not, but you have the option.
Yeah, no, the value is there, but it's, I think it's not showing here because it's not the workflow, right? You're showing the tool, right?
That's a good question. Let me follow up. I always thought if you clicked on this question mark, it would show you what the default value is. I'm not sure why it's not showing up there.
Okay.
But yeah.
So the tools that we have here for this workflow, they received the same default values that the tools you would run on your own computer. So when you download the tool and you open the parameter file already has a lot of those values filled with the default. All those parameters they already have are filled with the default values, and the same thing happens here. But I think it's not showing because it's those values are at the workflow, not the tool, right?
Yep.
Got it. Okay. Thank you.
That's another great question. So when a tool is running, since we're here, you can always click on View Stats and Logs to see where it is, and you'll see that it's working on MS Convert. So you have 2 tasks running, and each job has 25 files, so it has to convert them. So it's using MS Convert here. And whenever you see a sort of a horizontal line, you'll see the different steps of grabbing the inputs, working with the Docker, caching the Docker, and then the green is when it's actually running the job. And you can see now it's doing the MS Convert, and then you'll see it go into Workspace and then eventually into MS Fragger and then Peptide Profit. If you click on any one of these that's running, so this was complete, you can click View Logs and see, well, that's the MZML file. These are all the log files which are saved for you. And they're different for each tool. This doesn't, you can actually see the job tree. You can see, so it start to actually, not this one, but the Philosopher tools start to actually build a workspace. So what we came up with was, it's sort of expecting the workspace to be in one part. So what we're doing is we're passing a directory from step to step. And you'll see that later on if you want to look at it. Let me see. I just want to see if there's a real-time monitoring. Yeah. So if a job is running, you can click real-time monitoring, and it'll bring up this screen. All right, so I wasn't fast enough. It's already completed, but you'll see the live logs there. You can also click on instance metrics and see the CPU. So this was helpful when Felipe and I were optimizing it. So you'll see it's maxing out CPU, but not maxing out the RAM. So that's what I wanted to make sure that we're getting enough. We're using the CPUs because the less CPU you're using, the lower the cost of running the workflow. But we didn't want to run out of things like RAM or disk space. So we spent some time optimizing that as well. And so those defaults are built into the tool. Okay, so enough about that. So let's go ahead and everyone launch. Let me do a time check here. I'm going to pivot. Let's run this protein profit and talk about that, since we're talking a lot about workflows now, and then we'll talk about launching Data Cruncher, and then demonstrate the protein data commons import. So let's run another workflow because it's pretty easy to do. And so to run the second workflow, click on apps. And the second workflow is called protein profit. Now, normally you'd have to wait for the first step to finish. But for this project, we put all the results in the results folder. So let me show you how that looks. So on protein profit, click run. And this one's easy. It just has a workspace. It's required. And Felipe, so it has this one parameter. Can you tell us what this is? You're muted.
That's the minimum probability from the peptide profit validation. So peptide profit is doing the peptide validation. Meaning that it's looking at the distribution of all targeted and decoy peptides, and it's setting a probability for each identification that goes from 0 to 1, 1 being the maximum. That parameter here is specifying what's the peptide profit threshold that you want to allow to pass, and that means that if you increase the threshold, you're increasing the quality of the peptides you are allowing to be used for the protein inference.
Cool. So here's an example. This was an example, Paul, where we just wanted to show you one parameter. If you go in and edit the workflow, you can expose all the other parameters. So if we show, well, this one doesn't have a lot actually. It just has compared to the other tools. But you can go ahead and edit these. It's looking for default values. So these might not have any default values. Yeah, so it looks like just minimum probability has a default of 0.05. So we don't need to change it because that's the recommended value. So you can go ahead and leave that blank, and it'll grab the default. If there's no default, it just doesn't put the flag on the command line. All right, so this one's interesting. So on the workspace, so now the last step of the previous workflow is peptide profit. So at that point, it's built a philosopher workspace. So it's actually an archive of a bunch of files. Now, we did some optimizations here. So on the, if you go to select files, it knows that the input needs to be a tar file. But you'll see that there's, in this project, there's 46 tar files. That, yes, okay, 46 tar files. I broke them into 2 sections. One is for protein, so meaning going into protein profit. And the other one is going into the next app, which is the filtering app. And the reason we did that is to reduce the amount of data that gets moved around. And so let me know if this is confusing for people. Let's go ahead and let's filter down this list. So we have 46 files. Remember, we're doing an experiment with 23 plexes. We need to filter down for just the 23 plexes we want to use in protein profit. So the easy way to do that is just type in the free text here, type in protein, but spell it right. And so now I can look at the bottom right here. We see that we have 23. So does everyone understand that? I could have done something differently. I could have added tags or some type of metadata, but this was easy because the output is just for, it's the name of the CPTAC experiment and then for protein. And so at that point, select all 23 of these, save your selection, and hit run. Okay, and so this is not a batch test. This is a single job because protein profit needs all the information in one space. So this is a step where it's bringing everything together. And so what we did was we figured out what are the minimal amount of files that protein profit needs to do its job. And I guess it was designed normally to have everything available to it, even if it didn't need it. But here we're actually moving to do this analysis. We want to avoid moving data as much as possible. So we can let that run. The results
is
are
this a quantification step? Is this
Felipe?
quantification?
No, this is the step that does the protein inference. So here's what's happening. We have 23, or we have multiple experiments, right? Each experiment is composed by a different set of peptides. Now, what we want later at the final step is to have a protein expression matrix, right? But in order to do that, in order to have one expression matrix with a huge set of proteins that I can compare between experiments, I need to do the protein inference together, all combined. That will give me one set of proteins that it will be the same for all experiments, but they will be filled with different values for the quantification, because the quantification is relative to the peptides that were found in each experiment. So if you do the protein inference individually for each experiment, you will have different list of proteins, and that will be pretty hard to do any comparison. So what Protein Prophet is doing is gathering all peptide validation results from all experiments, pulling them together and creating one single protein inference list. And that list will be passed later again. It will be passed back to each experiment. And philosopher will use the list and the individual peptide list to do the FDR filtering and the quantification.
Great.
Thank you.
Another awesome question. That's a great point, because I'm glad Felipe's on the call. He's the expert for these tools, and he can explain it very, very well. So let's just keep running the workflows. I want to do another pivot, because I think Felipe led us into the next tool. So the next tool, next workflow is to do filtering and quantification. So remember, in your results directory in the files tab, you have all the results. So we don't have to wait for this to finish. I just want to give you an idea of what it's like to launch a tool, and so we can actually compress time here and zoom to the next part. So what is the next part? So the next part is the third tool, which is called Frag Pipe Filter Quant and Report. And it's actually, I don't know if you know this, but you can click on any of the apps and see a description. So we've written out this description here. This is an interesting one. It's doing the filtering first. So that filtering requires the output of Protein Prophet, as well as the workspace. And someone asked about annotation. This is where it's actually annotating the channels, right? And adding those here. So this is another sort of neat optimization we did. We're actually, the input's going to be an array of all the annotation files you want to use, and all the workspaces, the full-size workspaces that they're actually underscored for filter. And then some of the files will go to Philosopher Filter app, and then the annotation files actually are only used in label quant. So you can actually do this in CWL. So you can have one input and move some files one way, some files another way. This one takes about 45 minutes to run for each plex, 45 minutes to an hour. So let's go ahead and launch that. I'll back out again. So if you're on your apps page, find Filter Quant Report and click Run. Let's see what this looks like. How many parameters? So no parameter. So here we actually did, everything is hard coded. So that, that gives you some flexibility. Let's say you're working with your lab mates or your collaborators, and you want them to run the workflow, but you don't want them asking you how to fill out the parameters each time. So here, Felipe actually hard coded some parameters, not many. So it looks like PROT, which is the false discovery rate level. So he hard coded that, which happens to also be the default value. I'm trying to see if there's any. So here the PSM FDR is 0.01. So that's at the tool level. So that's why it says default 0.01 there. But yeah, and here's another default for MinProb, which has a 0.7. So I don't, I've never encountered so many parameters for these tools. They're quite a lot.
Yeah, usually these proteomics pipelines, they have a lot of parameters that you can tweak. So here what you're showing is all the different false discovery rate thresholds that you can apply during the filtering, because what Philosopher does is it takes all the peptides and proteins, divides them into layers that we call. So we have or levels. So we have a PSM, peptide ion, a peptide sequence, and a protein levels. And the FDR threshold, the FDR calculation is applied to each level individually. So at the end, what you see, what you're going to get is from Philosopher are different tables for each level, and all those tables are filtered at the corresponding FDR threshold.
Excellent. So the parameters that we don't have to change. So if I go back to editable, there's actually nothing that's editable in this. Yeah. So we made it easy for you. But later on, we can, in another webinar, we can discuss how you edit workflows to open up different parameters. So just think of it in the tool, everything's exposed. In the workflow, you have the option to expose only certain items to the end user, whatever you want. So you have that flexibility. So there's two inputs. Let's do the first one. Let's do the second one, which is protein XML. And this just one file. So if you go select file, now it should know to look for the correct file. I just forgot to do that part. So you can just type in prot XML, I think. Yeah, what's it going to bring up? Yep.
Just XML.
Yeah.
There we go. It's prot XML. Yeah. The first file over there.
Yeah, let's do the second one. So there's two. It should be smart enough, and we can upgrade the tool. It should just look for XML. So you see there's two. There's the first 21 and then the full 23 plex. So let's pick that one. So, because we're going to do the entire experiment. Right. Yes. So pick the second one there. And then this other one is absolutely required. So these are all your workspaces and annotation files. So let's do that. So this knows it's to look for two types of files, to look for the tar archive and the txt. So let's go ahead and run this on just the last two plexes again, because we don't want to use up all the nodes for this training billing group. On your own billing group, feel free to scale up as much as you want. So in the current workspace, we can actually, let me see if I did this. We can tag. If you click on tags, you can actually click plex 22 and 23. Yeah, so that's just, okay, so let's grab those two, but we still need the tar file. So another way to do this is to do, just start typing free text. So you can do 22 CPTAC. Has the for filter and the for protein. We're actually going to use the for filter, which is the entire Philosopher workspace. The for protein is just what Protein Prophet needs. So let's pick that. And then let's also grab the 23, which there. So if you saw what I did, I just updated the free text here. So I just typed 23 CPTAC. When you're done using the file picker, click save, and you should have 4 files. So we want to do another batch task example here. So to turn batching on, toggle it, turn batching on, batch by file metadata. Okay, and then same as the first step, choose plex or dataset name. Okay, and I'll give you 2 groups. So we have one group, the 22 CPTAC experiment, which has, if I can zoom in here, it has 22 CPTAC CCRC .forfilter.tar.gz, and it also has the accompanying annotation file. So the pipeline is going to know where to send each of those files, and it's the same way for 23. And I can show you in I have the template for the upcoming public project where I've run all 23 in one batch task. and it works really nicely. So the idea of a batch task is each group is going to get its own cloud node. So you can do everything in parallel. So let's go ahead and run that. And it's going to say you're about to run 2 tasks. Yep. So click run. And then it goes ahead and makes the child tasks. I usually hit refresh here, and then it shows you the 2 child tasks. So if you click on a child task, you'll just see the individual files here. Any questions about that? Great. So, all right, let's go ahead and just complete the workflows, and then we'll switch to interactive analysis and protein data commons, since we're on such a roll here. So now I want to show you the final app, which I did not include in your project. So if you go to your apps tab, you'll see that you only have 3 workflows, because I want to show you how to add apps from the public apps gallery. So we have to add TMT integrator. So to do that, in your apps tab, click on add app on the right side. And that automatically takes you to the the listing of all the public apps. There's also another way to get to the public apps gallery, which I may show you later, but this is the handy way. If you're already in your project, just you can add them directly from the public apps gallery this way. So how do we find Frag pipe? So there's 4 Frag pipes. We just need to get the one tool. So we can either search for Frag pipe, or we could look for proteomics categories, or look for the Frag pipe toolkit. The easiest thing, I always kind of revert to just typing in these search boxes because it's so handy. So I'm just going to type in Frag pipe, and then I see these 4 apps here, and you see that they're tagged with the lab that they came from. They're tagged with category and the version. So we're going to be always updating. Felipe and I are going to make sure that these get updated as we get feedback from you. So please provide feedback on the GitHub website about, I really wish the app could do this, or I saw a bug that's not quite right. Let us know, because Felipe did what's called third-party publishing. So we actually created these wrappers and then pushed them to the public apps gallery. So that's available to any of our collaborators that want to do this. And that's why you get this icon here, that it's coming from an external party. Okay, so which one do we need? We need a TMT integrator, which is on the bottom here. Frag pipe TMT integrator in QC. So just find the card you want and click copy. And then it asks, would you like to rename the URL? That's completely optional. So just click copy again. And then it gives you this banner, an app has been added to the project. So at this point, I close out, I hit the X in the upper right corner, and I see that I have a new app called TMT integrator. So this one has 2 steps. It has TMT integrator and TMT integrator QC, which is an R notebook. So let's run this, and then we'll switch over to R Studio. So this is just running the R notebook that we made based on the inputs. Will be the PSM files, right, from Philosopher Report, all of them that you have. So there should be one per plex, I think. So it should be 23. And then the database, the original database that we used. So then it makes multiple types of reports, different abundances, abundance by gene, abundance by protein, different matrices. So why there's two flows here. So this will actually give you all the report files. This notebook is just using the by abundance by gene. So that's why we have it going in 2 directions here. So when you're ready, click on run. And the PSM file should already be in the results folder. So once again, there's no app settings here. Is there any? So we haven't exposed anything to TMT integrator. So everything has been hard coded into the app itself.
Yeah, so TMT integrator actually works with a parameter file. That's why we don't have parameters there.
Yeah. Yep.
So you have to get that file and edit in the way that you need it.
Yeah, we can dive deeper into that. So I can show you, there's another project we call the commit project and the build project, which actually has, if you want to see behind the scenes, I don't know, I'll show you that time permitting. You can actually see each individual app, and it's interesting how we made that YAML file. So the YAML file is actually made every time you run TMT integrator, and there's certain parts that never change, and there's certain parts that do change, and we sort of did some CWL magic to make that work. So let's add the database. It should, yeah, so it pulls up the FAS database. So just pick that. And then the PSM files is going to be an array. So we use the file picker. It should know to look for so PSM files or TSV files. So we're going to get a whole bunch of PSM files. There's actually 98 here. So what we need to do is search for psm.tsv. So once again, I'm just going to use the text here, psm.tsv. And I have a sanity check because I can look that I see there's 23. So I'm going to select all 23 and save it. Save selection. And so this is not a batch task. It's going to use the entire array in one job. So let's go ahead and hit run there. And you'll see the report outputs on the right side when it finishes. I think this is about 30 minutes to finish this one. It'll include an HTML file that you can just open up right on the platform. So in the public project, we'll already have that there for you to see. But let's go ahead and launch into our studio. I think we're doing pretty good on time. Anyone need a break? You guys any questions about workflows? Because we're pretty much done with the workflows. So the idea is that the TMT integrator would make a bunch of matrices that you could then analyze and cluster in our studio.
Hi, this is Kritika again. I have a question. So we had run this pipeline, this entire workflow on a different cancer data set. Will we know as we're not experts in proteomics data, will we know what parameters to change, or can we use, assuming it's the same TNT10 experiment type, can we use the same parameters on a different data set of inputs?
That's a great question.
It depends. If your new data set, so here's the thing: different instruments will require different parameters for the database search. For the filtering, you can pretty much maintain the same parameters. The quantification might be adjusted, and the TMT integrator might also need some adjustment. That's because it depends if you are quantifying your data using MS2 or MS3 data. It depends on how many tags. For example, here we have a TMT10plex. You might have TMT6 or TMT11. So a few parameters might need to be adjusted.
Cool.
All right, thank you.
But Kritika, you can get in touch with me through that GitHub link that I pasted here on the chat. If you need assistance with that, I can help you.
Okay, very good. Thank you.
Sure, thanks. By the way, it's really exciting for me so I jump back out into the platform dashboard, and that's where you can see the overview of all the projects you're in. So I'm in everyone's webinar project. So it's really exciting that I see everyone has made it to the last workflow already. So that's great. So let's pivot to the other type of analysis, which is we call data cruncher, which is just a term for running RStudio or Jupyter Lab in a container inside your project. So it's your own private instance of either one of those two things, and you'll have access to anything in your project. So it's like you're still contained in your project, but we can go ahead and analyze the data that's in our results folder. So to do that, to get it started, and it loads up pretty fast, so we can just talk while it's loading. There's two ways to get to it. In your project, if you go to the dashboard, and I'll just show you this way because it's the shortcut. On your dashboard, I clicked on it again. On the bottom, you'll see there's two types of analysis. You have tasks and data cruncher. If you click on data cruncher, you'll see any data cruncher environments you've already created. So I made one called RStudio Frag Pipe Data Exploration. So go ahead and click on that. By the way, if you wanted to make a new one from scratch, which you're welcome to, you would go to interactive analysis and then pick what type of environment you wanted. So click on this data cruncher. And the idea about this, so it's not starting from zero. It's actually starting with all these files in the workspace. So the idea is it's its own container, and you can go ahead and copy this to another project or fork it and change it a whole bunch of stuff, a whole bunch of times. And you can actually preview what we have in the workspace, which includes Felipe's notebook, as well as some config files. So go ahead and hit start. And this will take 2 to 3 minutes to load, 2 to 3 or 4 minutes. It's going to take just a little bit longer because what it's doing is provisioning an instance on Amazon, because this is an Amazon project. We also have Google projects. If you're in a Google project, it would make a Google RStudio for you. At the final step, it's actually going to load a library that's required for Felipe's notebook, and it automatically loads that up for you. So this will just take a moment. But yeah, any questions about RStudio? Let me just show you while this is loading, let me know if anyone had problems finding the start button. Let me show you the other way to launch it. So if you're in your project dashboard, on the right side, you click on interactive analysis. And then this actually gives you the different types of apps. So we're always adding different things here. There's a genome browser and a variant browser. Data Cruncher is Jupyter Lab in RStudio, so you open that. And then this actually gives you an option of starting the environment you already have, or creating a new analysis. So I can show you how to make Jupyter Lab. Let's say you create new analysis, and then you pick your either environment you want, and give it a name. So the idea is inside the environment here in the workspace, you would keep all your scripts because you can edit them in real time, and you can move the environment around from project to project. The files should remain in your files tab in your project because those are larger. And you'll have access to the files inside the running environment. And then you can pick on what environment setup you want. We're going to increase the number of environmental setups here in the future and have different options available for you. There's a machine learning environment as well, but normally I just pick the default. You click next, and then you can actually specify what compute you want. And suspend time is always left on. And then you could, although one user was keeping it on 24/7, and I had to say, let's think about, turning it off when you're sleeping at least. So I usually keep it at 30 minutes or 60 minutes. So when it detects that there's no CPU running, it'll go ahead and shut it down. And it does it pretty safely, actually. It saves even if you haven't saved a script, I've noticed that it'll go ahead and save a copy of it. But please save early and often. And then you just click start, and it provisions the instance for you. So let's see where we're at.
Is the way you index the files in the buckets same for both Amazon and Google Clouds?
It's a good question. I think it's generally the same. It's generally the same. It's sort of like, what is it called? Like invisible to you. The only difference, when you make a project, it's either going to be a Google or Amazon project. So that, and you can't switch it later on. But you don't see how the things are indexed. Everything appears exactly the same. The only time you may notice a difference is when you're running an analysis, or you're running a task, and you go into view stats and logs, you'll see Google names of instances. So they name their instances different, and they actually have different levels. I'm most familiar with Amazon, but Google has a different way of naming their compute.
Okay.
Okay, so let me see. Let me go back up here. I don't want to actually use JupyterLab. We're going to focus on RStudio. So mine's running. So what I do is I can click on open in new tab. Did it already open? Yeah, okay. If you're on the screen, it'll automatically open for you. Oh, I see. So this, if it's doing this, what you see on my screen, it's actually loading the final library. And this only takes 30 seconds. But this is a good test, especially if you haven't, used RStudio on the platform in a while. Let us know if you couldn't open it up, and we could help you troubleshoot your browser settings maybe. So what else is on the agenda? So we have walking through the notebook here, and then looking at the PDC website, which will probably end on that. Don't know why it said that. Okay, so it automatically loads up because of the R profile here, which I added. You can automatically have files load up for you, and I've included the TMT integrator QC or Markdown file. So if you're not familiar with RStudio, there's different window panes. So on the left side you have your script, and on the right side you have your files or your environment, and then the bottom right, the console. This is actually slightly different than the default. This is the way sort of I prefer to look at it. But anyway, in the R Markdown file, there's a button called knit. So we're going to knit this report. So if you just click on knit, it should take care of the rest. So it's going to actually run all this script, and it's going to make the HTML file. And that'll open up in a moment. And what I wanted to demonstrate here is that this is the exact script that we're running in the tool, which is called the QC tool. But you can actually go ahead and take the script and modify it. So Felipe, do you want to walk us through this?
Yeah, so here what David is showing is a few plots. This is just actually an example of how to go over the normalized quantification for each sample and see if things looks okay. So here, for example, David took a slice of the whole data. He's just showing us what, about like 20 samples.
Yeah, I think first 20.
Each column here, each box plot is a different patient. We can see the distribution of the normalization or the intensities across all the patients. The same thing in the box below. So this is just the density plot showing the distribution of all intensities from all patients. So this is just an example of how you can quickly go from the data reports or the data output to something like a report that you can download and share with others.
Yeah, and there may be like certain samples that you're really interested in. So there's a part in the script above there. If you're an R person, you could, here I'm just grabbing the first 20 here. You can dive into this, the first 20 channels. You could change it to the next 20 or grab specific names. But I think the heat map is pretty interesting, or at least we're getting some clustering here, right? So there's like 3 main groups.
Yeah, part of these samples are tumor samples, and the other part is normal adjacent samples. So we don't know which one is each one because the labels here that we use do not indicate that, but we can clearly see that we have a distinct separation between some of those groups.
Any questions about RStudio or Data Cruncher in general and what the capabilities are? They're always making improvements to this, so stay tuned for that. It should be in the future a lot easier to save your environment.
I have a question.
Of course.
Can I install different packages than the ones used here? Or?
Absolutely. Absolutely. So this is your own sort of private domain. You can install whatever packages you want, either via the Ars console. Let me see. I put it down here. So we're in the console. You're going to install packages here. You also have terminal here, which you have sudo, so you can go ahead and install anything. They're not persisting from session to session. So that's something that's on the roadmap to make that a better user experience. So if you do install something, when you boot up the environment, it'll be there for you. But a lot of times I'll just make, that's what I included here. I included actually like and I learned how to do this just for this webinar. If you're an R person, there's a .r profile, which you can go ahead and just type in the packages you want to install, and it'll automatically install those when you start it up. Okay.
Does the R profile persist from session to session?
It does, yes, and it absolutely does. And it took me a while to figure this out. But I think this will be really handy for a lot of users because, by the way, that is the question. There's two questions for Data Quencher I always get. One is, can I install packages and make them? How do I make them persist? And the second one, which I'll mention right now, is how do I find my project files? But yeah, I think this is a great solution to automatically have certain things install, and it's just part of the boot up process. So files. Okay, interesting. Okay, so let me show you about the files. I have to show you in terminal down here. So where are we? So we're in SB Genomics workspace. So imagine, this is your own private server. Anything in your workspace will be saved in the environment, and then you can move it to another location. But anything in the files tab, so we have the workspace, but anything in the files tab is under project files. So if we go to project-files, we can see everything that we had here. The CPTAC raw files. So these are all the raw files, yeah. And anything you save back to your project will be there. So let's say you wanted to access here. Yeah, so in the results folder, I broke it down into the different steps. So there's convert, filter, protein profit, TMT integrator. TMT integrator might be interesting to explore. There's a bunch of different TSV files. So I don't know if you can see that. Yeah, we're going to critique. Yeah, we want to distribute the webinar. I think Dan's going to help us do that.
Thank you.
But yeah, so there's different abundances. So there's group by gene, group by peptide, group by protein, different ratio things. So those are all there for you to ingest into R and make plots on. Okay, so how did we get this data? I need to make sure that I give credit to Proteomics Data Commons and give the credit where credit is due. So if you go to proteomic.datacommons.cancer.gov, so this, all the data we use today is open data. So I think it would be really neat if you guys would do this flow for a different disease type. So we did, today we did kidney cancer, and there's 119 cases. But you can pick a different one and do this import and just use the same projects. I think we can, if you want to use the same project, and we'll see how it goes. Okay, so we have four.
The parameters would be different, right? Is what Felipe said. If we use a different cancer type, the parameters would be different as well.
Is that true? So yeah, I think, so first step, reach out to Felipe and do what he says, not what I say.
Yeah, so it depends. I did processing of several of the CPTEC3 cohorts so far, and most of them share the same parameters. But yeah, one of other cohort, they need you need to do some adjustments. So again, it depends on what kind of data you're processing. You need to know if they use the same instrument, the quantification was done the same way, and things like that.
But so maybe if there's a different cancer in PDC which has the same parameters, we could try that out.
Yes.
That's a good point. We should find that out for you.
Yeah. So if you want to try another CPTEC3 data set, the parameters will be pretty much the same.
Okay. Thank you.
Oh. Yes, I think people were trying to help me do this. Yeah, we're going to use the Files tab to import this. So what's the next step? So we have export study manifest. Oh, yeah, files. Sorry. Okay, so we have we want to actually get the raw files. How many files do we have? That's too many. No, that's it. That's the MZML files. I need the that's the right number. It's just the wrong number of files. So kidney. Yeah, so you click on the study you want, and then you click on the type of data category. So there's raw MZML, has some PSM files, a lot of PSM files already. So we want to actually click on the raw files. Okay, and then we click all the, so we get a, we need to download all the files. So we need to make sure that we export the manifest in CSV. Let me try this.
You have downloaded those files already?
Yeah, so we downloaded the CSV, and then the next step is to go to your files tab, and finish the import, which I've already done for you, but this will be how you would use it for another project. You go to add files, and then import from a file manifest. So there's different ways to get data, but if you're working with PDC data, you go to the import from file manifest, and then the default is the PDC, but there's different options here. And then you select the file manifest and go ahead. I'm not going to import it because I already have the files there, but you import it, it's really quick, and you have the data there available. All right, we're just about time. We have 6 minutes left. Any other questions?
For the data cruncher, the last time I tried it, I think the output folder was the same as the input folder. Is that still the case?
So let me talk about that. There are two different folders. So there's the input folder is what's called project files. So those are all read-only. If you do want to save files back to your project, the current solution is to save them to the output files folder. So any like generated data that you don't want to just keep in the workspace, you want to actually move it to the output. So let me just make a fake file. Like if you had a test.csv. So this is in the output files, test.csv. As soon as I hit stop, it's going to be a file that was created by this analysis. So it's, as soon as it finishes saving, you'll see it there.
Okay, thank you.
Yeah, so here's it. So there's two concepts. So there's the workspace. So these will always move around. If you decide to clone this and copy it to another project, you'll always have the workspace available, everything in the workspace. The output files are just things that are saving back to your files tab. So if I go to my files tab, I'll see this file, test.csv. Of course, it's blank because I just made it.
Right. Okay. Thank you.
Time flies. It's 2 hours is interesting because it's for what I want to show you, maybe it's kind of short, but it does fly by. I want to thank you all for your attention. And the way we set up the projects is kind of unique this time because I'm in, I saw a TMT integrator completed. That's awesome. You feel free to go ahead and reach out to me in any of your projects. Just make a note. And you'll be able to see a conversation. We can have a conversation right in your project about any of your files. This was the first one that actually finished. So let me just look at that. So the gene, so you have the gene report, which is HTML, and then of course all the abundances, which are TSV. Whenever you see the histogram, that there's a visualization available. So you can open that in a new tab and you could see HTML displays really nicely. It's the same thing as in our studio. And then the gene abundances, if they're below a certain number of megabytes, they will display in a nice preview. So you can go ahead and, sort. You can even search. But if it gets too large, like really large files, you won't be able to do that. You'll need to open RStudio. Great. Any closing remarks, Sai?
No, I think this was great. Thanks so much, Dave, and thanks for your time, Felipe, for joining us and making this workshop possible. And I want to thank all of the attendees for staying for the entire 2 hours and trying this out. So if you have any questions, like Dave mentioned, reach out to Dave. He's available for office hours tomorrow. It's the link to join the office hours is in the website. But you can also email, like he pointed out. So thanks, everybody.
Thank you. It's great. Great job.